Welcome everyone to the AME seminar. We are so excited to be hosting Matthijs Kox this seminar. And of course, we wish that it was in person. We'll have to plan for that because he is in the Radboud University Medical Center in Nijmegen in the Netherlands. And we have been reading about and admiring his work for many years now. And I believe it's fair to say that he is the person most responsible for creating, putting the Wim Hof method on the research map, getting researchers interested in it, and creating a small but fast-growing field on people interested in hormetic stress, hypoxic stress, and in particular the Wim Hof method. So his research portfolio is fascinating. They are translational studies on immune response in critically ill patients with a focus on treating immunoparalysis in sepsis. So he's dealing with, really acute conditions in inpatient wards. He developed a unique model to study and treat immunoparalysis in humans in vivo using repeated endotoxin administration in healthy volunteers. Is that like a hormetic stress response?
Yeah, it's an acute model of inflammation, and it also elicits stress actually. So you see also cortisol and other hormones increasing. Yeah.
Wow. In terms of his Wim Hof studies, well known, Wim Hof is known as the Iceman, and he claims that he can voluntarily modulate the autonomic nervous system and the immune system through his methods. And the best study was the PNES study we'll hear about by Matthijs. And since then, there's been other studies that have built on it. So I won't take away the punchlines, but I will say we are absolutely planning on continuing on studying the method and some of the measures and outcomes that Matthijs has been focusing on. He focuses on the entire spectrum from molecules to population work, in vitro work, animal experiments, healthy volunteers, and patients. He supervised over 40 biomedical research internships and 25 PhD students. All right. And we're just happy and hope we can collaborate in the future. That's been a goal of ours for our studies. And we're actually, we'll talk about that later. So welcome. You have this amazing group of researchers here. We have some of our research staff, postdocs, faculty, psychiatrists, psychologists. And so it looks like we're doing screen off, but at the end, we'll have plenty of time for Q&A. We'll do screens on. Thank you, Matthijs. It's 9 p.m. for you, so we won't keep you past 10.
Thank you very much, Elissa, for this very nice introduction. I will start sharing my screen, hopefully. Okay, great. So, thank you all for joining. It's really nice to have you all virtually online from the other side of the pond. So that's also an honor for me actually to speak to you. So thank you very much for joining. And today I'm going to tell something about voluntary control over your immune system and whether that is possible or not. What my talk is about, and this is no surprise because Elissa already told a bit about it, rightfully so. It's about this guy Wim Hof. He's a Dutch individual, and he's better known as the Iceman. But the other side of my story is about the immune system. So it's about the interplay between these two. Between this guy actually and the immune system that my talk will be about. Now, first, a bit about Wim Hof. I'm sure you know most of it, but he has several former world records and also current related to cold. So he can spend almost 2 hours immersed in ice like this. He has the fastest half marathon barefoot within the Arctic Circle. At minus 20 to minus 30 degrees centigrade. And he swam more than 50 meters under ice, which is also quite impressive. But what was the most appealing to us, because I'm an immunological researcher, as Elissa said, at the ICU. So normally we study critically ill patients who are very ill with dysregulated immune response, actually. So sepsis is my main focus. And as Elissa also said, sepsis-induced immunoparalysis is the particular focus, which is a severe dysregulation of the immune response in these critically ill patients. But Wim Hof, at the time I was actually a long time ago, I was doing my PhD, and that was about the interplay between the autonomic nervous system and the immune system. And Wim Hof actually kind of came to us and said, okay, I have developed a method that allows me to modulate my own autonomic nervous system and immune system. And that kind of triggered us, of course, because we were researching that exact topic, and this was kind of like an outlandish claim as well, because both the autonomic nervous system and the immune response are both autonomic. Actually, it's not something that you cannot like voluntarily influence. At least that's what was in the textbooks. So first of all, at first we thought, okay, should we do this? Should we take the plunge and study this like this media figure? We were quite reluctant, I have to say, honestly. But then we really researched a bit on what he had achieved and what kind of records he held, and we thought, okay, this is a special guy, and let's give him the chance. So we tested his claim. Before I start with the studies, actually, I want to tell you a small bit about the immune system, which you also might know already, but still. The immune system, of course, recognizes invading pathogens such as bacteria, viruses, yeasts, fungi. And this is, for instance, a schematic picture of a bacteria, in this case, the E. coli bacteria. And that the immune system recognizes these pathogens via conserved receptors known as PRRs, such as TLRs and RIG-I receptors. And that leads to kickstarting of the immune system, of the innate immune system. And one of the key hallmarks of this immune response that is generated is the production of large quantities of inflammatory proteins known as cytokines. And one of the primary goals of these cytokines is they are the messengers of the immune system, so they also attract other immune cells to the site of infection and instruct them there to do their job, actually. And of course, the goal of this immune response is to clear the infection. And as I said before, it's an autonomic process. It's not something that you can say, okay, now I want to willingly like activate or suppress my immune system. It's something that goes involuntarily, actually. So the immune system is so it's very important, and without it we wouldn't survive for a day, I think, or I know actually. But sometimes the reaction of the immune system is also can be too vigorous or is inappropriate, and this causes actually damage, some sort of collateral damage to tissues. And one of the prime examples, which I work on most of my time, is sepsis. So it's also known as blood poisoning, and that's a very severe condition with a mortality rate up to 30%. And it's actually in the US, for instance, also in the Netherlands, is the most expensive condition there is. And it accounts for like 5% of the total healthcare budget in the US. Trauma is another example where you have where you can have excessive immune activation leading to tissue damage. But there are also instances when the immune response is inappropriate. Actually, it's maybe not always too vigorous, but inappropriate, and that's the case in autoimmune diseases such as rheumatoid arthritis or Crohn's disease, where you have like an immune response directed against your own tissues, not against a pathogen, but it's like an error in your immune system that it attacks your own cells and tissues, and of course that's detrimental. I talked about Crohn's disease already. And the therapy in these patients is mostly to suppress the immune system, of course, using drugs. So well-known are corticosteroids, for instance, but also biologicals such as anti-TNF are quite successful for rheumatoid arthritis and other inflammatory conditions. So let's get back to this bacteria, because I want to tell you something about studies into the immune system and how we do that. And we extensively actually make use of endotoxin. It's also called LPS. I will use these words kind of interchangeably because it's the same thing. And the toxin is a constituent of the cell wall of gram-negative bacteria, such as the E. coli bacteria. And that is exactly the molecule that is recognized by the immune system as being foreign and as being pathogenic. So then the immune system knows I have to mount an immune response. And it's a very strong immune response. It's a very potent activator, therefore, of the immune system, and therefore it's widely used in research into the immune system. And normally that's mainly done in that's mainly done in animal studies or maybe in vitro in cellular models. But we in Nijmegen we do something quite special. We inject this LPS, it's highly purified, into healthy volunteers. And what happens then is, as you might expect, these volunteers they generate an immune response. And what happens then is all the classic inflammatory symptoms that you all know of, such as a fever, an increase in heart rate, an increase in flu-like symptoms, such as shivering, nausea, headaches, muscle aches, all kinds of things, and a decrease in blood pressure. And as you might appreciate from these slides, you can see that the response is quite acute, but also quite short-lived. So 8 hours after injection of the LPS intravenously, the subjects go home happily ever after, and they have no symptoms or complaints anymore. And it's a perfectly safe model because it's not a real bacteria, it's only a dead part which cannot replicate, and is actually cleared very quickly from the circulation, but the response lasts for a couple of hours afterwards. And what is for us also very important is that we can measure very objectively the immune response using a cytokine assay. So as you can see here, two of the prototype pro-inflammatory cytokines, TNF and IL-6, they also sharply increase after LPS administration, and then also sharply decline afterwards. And after 8 hours, they're completely back to normal levels. Apart from these pro-inflammatory cytokines, also anti-inflammatory cytokines are produced kind of like in a feedback loop to dampen excessive inflammation. And IL-10 is actually the prototype of the archetypal anti-inflammatory cytokine that we measure. And so we subjected Wim Hof to this model of inflammation, and we call it the human endotoxemia model, and that's the LPS administration in healthy volunteers. And here you can see a picture. It's quite a long time ago, and we measured basically like everything we could measure on that day, because of course he was and is a special person, and we wanted to make the most of it. And I will not go into much detail on the results, but what we noticed was that he experienced virtually no symptoms, and he had very low cytokine concentrations in his blood, because we compared him to a group of over 100 volunteers who were challenged with LPS before. And we also saw a sharp increase actually of adrenaline in the blood. So, and that points towards activation of the sympathetic nervous system, as you might know. This is a graphical representation of the nervous system, with at the top the central nervous system and the peripheral nervous system. And as one of the subdivisions of the peripheral nervous system is the autonomic nervous system, which is the system which also acts involuntarily. So it's not something that you can control, not like when I say I want to put my hand up, I can put my hand up, but the autonomic nervous system is not like that. You cannot really control it. It controls, for instance, things like your heart rate, your digestion, and those are not things that you can willingly influence, at least not according to the textbooks. And today you have actually two parts of the autonomic nervous system: the parasympathetic and the sympathetic division. And today I will talk most about the sympathetic division, because there we saw the major effects of the method of Wim Hof. And as you also are quite aware of, I think, is that the sympathetic nervous system is involved in this fight or flight response. So if you are like robbed on the street by a burglar, then this system is activated, and you either run or fight this burglar. And it's not, and again, that's not something you can really control. It's most of these things go automatically. So also this, the name says it already, it's involuntarily. And what's important to realize is that adrenaline, which we saw that was increased to a large extent in the study in Wim Hof, it's the neurotransmitter of the sympathetic nervous system. So this is the signaling molecule that indicates that there's activity of this part of the nervous system. And what is interesting about adrenaline is that we know already, also before our studies, that it can exert immunological effects. So this is also a study where they injected LPS into healthy volunteers. It's not by our group, but by another group. And in conjunction with LPS, they administered either placebo or adrenaline. And as you might appreciate, the group that received adrenaline next to LPS, they had much lower TNF, so pro-inflammatory responses, compared to the group that received placebo instead next to LPS. And so actually this neurotransmitter, if you administer exogenously, so this is just by an infusion pump intravenously, an administration of adrenaline, and then it dampens your immune response. But in the studies we did, it's endogenous production to such high levels that you can also exert this response, and that's quite special, I think. So adrenaline inhibits this production of pro-inflammatory cytokines and puts the brake on inflammation. What's important to realize in the study that we did in Wim Hof, and as you are all scientists, you know this, that it's an NS1 study. So it's a study, and with an NS1 study, you cannot really do like statistics, because you can only compare it to a historical control group and then say something about that it's lower or higher or a bit different, but you cannot do real statistics on it and say this is really happening. It could also be the case that Wim Hof is just a genetically different, and that therefore he's a very low responder to LPS, for instance. That could also have been the case. So if we would have to really have to prove his claim, we would have to test groups of which half would be trained by him and half would not. And then his next claim came into play. He actually said, this is a newspaper in Dutch, but I translated this headline: anyone can do what I do, and I can train anyone also within a short period of time, like within a week. So that opened up the opportunity for us to do a study, a follow-up study, in groups of subjects. And we actually wrote a grant proposal to the Dutch Rheumatology Society, and we got the grant to do this study in 24 healthy volunteers, and 12 of those were trained by Wim Hof, and the other 12 were untrained controls. So they just stayed at home, did no training, and went about their normal lives. And all these volunteers again were injected with LPS, as I've shown you before, to test the immune response. And the trained volunteers practiced the breathing technique, which I will explain in a bit in more detail during the LPS experiment. It's important to realize that the cold exposure part and the meditation all took place during the training period, but not during the experiment itself. So when they were with us at the research unit and we injected them with LPS, they did the breathing exercise, but were not subjected to cold or did not practice the meditation. Oh, I hope this works. Yeah. So this is the volunteers were actually trained in Poland in part and partly in the Netherlands. And this is a short clip of the training procedures in Poland. So they did all kinds of things. Of course, the exposure to cold was a large element of the training. So they stood in snow for up to 20 minutes and did all these exercises. And I think they also lied in snow for up to 30 minutes. And as you can see, it was quite cold there. I of course was also there to do a bit of supervision, also to look what the training exactly was actually, because beforehand we did not really know that. We just gave Wim Hof kind of the free hand to do his training, and we observed. So they did also after the snow line, they did the rewarming in the sauna. And this was also quite impressive. This is like a frozen waterfall, and you can see the ice in the icy lake. And yeah, these guys, they all could do it actually. So after a few days, and already on the first day, they could do a lot, and it became increasingly impressive what they could achieve. using all the training and also the motivation by Wim Hof. So this was really impressive to experience that. We took all kinds of precaution measures, of course, with blankets and an AED and everything but nothing was, we didn't use any of those, luckily. And the last day of the training, so it was 4 days in Poland, they hiked up the snowy mountain. It's actually the highest mountain in Poland. And it was a blizzard at the time, and it was really cold. So I also went up there, but it was minus 27. As you can see, these guys only wear shorts and shoes, but no shirt. So and they really flew up this mountain, and as you can see here, the weather conditions were quite horrible, actually. And at the top there was this weather station of the Polish meteorological service, and there we could have some tea and warm up. But these guys were so enthusiastic and motivated that they didn't feel the cold at all. But you can see that their legs were icy, and it was really quite impressive to see again. Yeah, and of course they also did the meditation and breathing techniques, but those are a little bit harder to catch on camera. But yeah, you probably know this also from your own studies that what happened here. And they also did some yoga and strength exercises during this training procedure. So when we came back from Poland, we finished the training by some additional days in the Netherlands at the sports center of the university, and we also had some ice baths there with the subjects. And then the experiment day was there, and I already said that I would show you a bit more about the breathing technique. And as you may be aware of, it's cycles of hyperventilation followed by breath holding. Hopefully it works. No, it doesn't. Let me check. I will do it like this. So I hope you can all see it. So this is one of our subjects, and as you can see, it's connected to this patient monitor, and you can click on you can see the purple figure is quite, that's the one to watch actually. It's this O2 saturation, and if he first started with 95 seconds of hyperventilation, and of course the saturation then is like at 100%. And he said, okay, when I stop breathing, I will put up my finger, and then you can take a blood gas sample actually to measure the acidity and all these things in the blood. And as you can see, he has a pH of 7.75, which is really impressively high actually, so very alkaline. And then and a very low CO2, of course. And then he held his breath for 2 minutes. And he turns a bit blue, as you might appreciate as well. And then he said, okay, I'm going to start breathing again, take a sample. And as you can see, the O2 saturation is 55, and it actually went down to 46% in the end. That was the lowest we recorded before he started breathing again. So, and normally, like our nurses on the ICU, they get a bit anxious when it's under 80, now under 90 actually. So this is a really low level. As you can see, it's now 46, and that was the lowest point. So that was the breathing technique. So of course, we measured adrenaline again, and this is the control group where you see a totally normal response. Due to the inflammation, adrenaline also increases. That's what we always see. But in the guys that were trained, we saw a much more strong response, which happened much earlier. And the blue bar indicates the period in which they performed the breathing exercise, and you can see that it immediately goes to very high levels, and it stays relatively high during the practicing of the breathing techniques. And it's actually higher than bungee jumpers, and they studied this in Amsterdam actually. where they just took a sample before someone bungee jumped for the first time, and they actually found lower levels of adrenaline than we did. So we were quite happy with that. So then to the symptoms, the flu-like symptoms and the fever. And these are the control group data again. We see a totally normal response with sharp increase in symptoms and a fever of about 2 degrees. But in the train group, the symptoms were more than halved actually, and also the fever was attenuated in this group. So that was also a strong effect there. And then if we look at the cytokines, this is the anti-inflammatory cytokine IL-10 in the control group. On the left is the time concentration curve. On the right is the area under the curve of this time concentration curve shown on the left. And as you can, this is again a totally normal response in a control group with a peak at 3 hours. But what we saw in the train group is that the adrenaline increased very sharply early on, much earlier than what we normally see. And it also peaked earlier in this group, and it went to very, very high levels, so 3 times higher than what we saw in the control group. If you then look at the pro-inflammatory cytokines, so TNF, IL-6, IL-8, we see that those are basically halved compared with in the train group compared with the control group. And we also did a lot of correlation analysis, and we actually could tie all of these together. So we saw that the early increase in adrenaline led to the early increase in IL-10, and the IL-10 was related to the dampening of the pro-inflammatory cytokine response, which kind of all made sense. Because, as I said before, it is known that adrenaline can dampen the immune response, and I didn't show you that, but it's also known that it can induce IL-10 to a very large extent. So, in summary, in this PNAS study, we saw that these trained volunteers showed extreme fluctuation in acid-base balance and short but deep periods of hypoxia, or short periods of deep hypoxia, actually. We saw a very rapid increase of adrenaline in the blood to very high concentrations. And we saw adrenaline induce strong and early increase in anti-inflammatory IL-10. And again, this IL-10 in turn mediated the inhibition of pro-inflammatory cytokine production. And we also saw that the symptoms were halved and that the subjects had less fever. So this was all kind of good news. And in conclusion, we could say that this method allows for voluntary activation of the sympathetic nervous system and attenuation of the immune response, or the acute immune response. And you can also frame this voluntary activation of the sympathetic nervous system as a kind of controlled stress response, I would say. So this is something that it is an acute stress response, but it's kind of in a controlled manner. So how we thought, okay, how to proceed with this method or training program, and we thought, okay, if we want to mold this into a real treatment, we have to do a couple of things. You have to show that it's reproducible, and also importantly, which of the elements of the method cause the observed effect, because you can imagine that not all patients are able or maybe even willing to go into ice baths, and also the breathing exercise might pose difficulties for some subjects. Not all people like it. Actually, most people do actually the breathing exercise, but not all of them like it. So, and also the hypoxia could, for people with cardiac disease, could also be potentially harmful. So it would be nice to know whether only one of the elements, for instance, would be sufficient, or that you really need all of these elements. So these are the 3 elements, of course, which you probably know. It's the breathing, it's the cold exposure, and the meditation. And we did a lot of literature research, and we thought, okay, we think that most of the effects can be ascribed to breathing or cold. We had so there were in the literature some indications of that. And for meditation, it was a bit all over the place, actually, the effects of meditation on inflammation. And also we consulted with the participants of the first study, of the PNAS study, and they were a bit vague about the meditation. They said, okay, this was something that was not really clearly defined during the training, and sometimes we just slept, and it was not really something that they paid a lot of attention to. So we thought, okay, let's focus on breathing and cold, also because these endotoxemia studies are really, really labor intensive and expensive. And we wanted to, see if we would do a full factorial design with 3 interventions, we would need very many subjects and a very large study. So we focused on this. On these 2 methods. And to dissect the method, our research question was, what are the individual and combined effects of cold exposure and breathing exercise on the immune response? And what we also did is use an independent trainer instead of Wim Hof, because if you would really want to mold this into a treatment for patients, then of course it would not come in very handy to have Wim Hof around all the time. So someone else should be able to do the training as well. So we had 48 healthy volunteers in this study. Again, we had a control condition, which the subjects did not receive any training. We had kind of the same condition as in the PNAS study with the combined intervention, but this time without the meditation then. But we also had the separate condition, so only cold exposure training and only the breathing exercises. And again, of course, we use the LPS injection model to measure the immune response in these subjects. And this, these are some pictures of the training. We didn't go to Poland this time, and it was not that cold, but we still, we got some snow, we got ice. So they did. They also, they did they had a pool which we cooled with ice packs. And we had the independent trainer doing kind of the same exercises that Wim Hof did with the subjects in the PNAS study. So we try to copy that training procedures as closely as possible. And so these kind of things were also done in Poland, and we try to replicate that as faithfully as possible. And at the last day, so we increasingly made the pool colder with a lot of ice cubes. And at the end, they did all these exercises, also comparable to what was done in Poland in the PNAS study. So we try to follow that as closely as possible. So again, adrenaline, of course, that was one of the important endpoints here. And this is, again, the control group. It's a different time scale a bit, but it's the same response as you normally, as we saw in the PNAS study, with an increase at 1.5 hours after LPS injection due to the inflammatory response. In the cold only group, we didn't see any effect actually. From that we thought, okay, it must be the breathing, and indeed it was. That we saw in the breathing group and in the combined group that there was this large increase again of adrenaline to very high levels, which we did not see in the cold group and in the control group. So from that we could conclude that the adrenaline at least was induced by the breathing exercise and not by the previous cold exposure. And in terms of symptoms, this is again a control condition. We did see a large reduction in symptoms in the cold only group. So cold exposure training appears to influence these flu-like symptoms elicited by LPS administration. In the breathing only group, we saw that the peak was comparable to the control group of the symptoms, but they declined much faster than in the control group, as you might appreciate from this figure. And the combined group was actually right in the middle of the cold group and the breathing group. So we saw a less intense peak of symptoms and also a swifter decline compared with the control group. And this is a summary of the cytokines in the blood. I will not show you all the individual graphs, but as you can appreciate from this slide, is that the cold only group, we only saw a small reduction in MCP1, but no effect on any of the other cytokines. In the breathing only group, we saw a reduction of nearly all pro-inflammatory cytokines, but no effect on IL-10, on the anti-inflammatory cytokine. And in the combined group, so which is most comparable to the PNAS study, we saw actually a decrease of all pro-inflammatory cytokines, and again this increase in anti-inflammatory IL-10. So the strongest effects was clearly seen in the combined intervention group. So the conclusion of this study was that if we dissect the method, again, and this is important, I think, to stress, that we see that we can reproduce the findings that we found in the PNAS study. So The effectiveness of combined cold exposure and breathing exercises on the immune system were clearly noticed again. And that the breathing exercises have the largest impact on the immune system, but that cold exposure training potentiates these effects to a significant extent. And that importantly also that the cold exposure training alone attenuates the flu-like symptoms. And these symptoms were interesting to us as well, because we thought, okay, are these just attenuation in symptoms? Are they due? Is it only like dampening of the immune response that is responsible? Maybe not, but because in the cold exposure group we also saw less symptoms, but no real effect on the immune response. So we wanted to investigate in the same subjects, in the same study actually, whether there was a direct effect of the method or the different elements of the method on pain perception. And we did that using a standardized battery of tests actually. And in this battery we tested pressure pain with this pressure transducer, which we put on different parts of the body, and then asked when it became painful, and then we read the value in Newton meters. We measured electrical pain, so we put on a couple of electrodes, and then current started flowing in increasing levels, and then when it became painful, the subjects had to release the button, and then we could read how many milliamps were flowing. And we also did the cold induced pain, so the CPT, the cold pressure test with the ice bucket, which is quite well known. And in this study we also did this not only during the endotoxemia experiments, but also in a separate set of experiments where they only did the breathing exercise. And what we noticed was that during the breathing exercise, we saw that the pain threshold for electrical pain was increased. So actually, the subjects could take more current, more pain, more electrical current before it became painful, compared to before they started with the breathing exercise. But we saw no effects on the pressure pain or the cold-induced pain. And actually, this effect lasted, because also 4 hours after cessation of the breathing exercise, we still saw an increased pain threshold for electrical pain, and so the pressure pain and the cold-induced pain were not affected, but that was also in the acute phase. So we did not expect that here again. But the electrical pain, we saw a significant effect there. And during the endotoxemia, this is the first showing what the training itself did. So this was we measured these pain types before the subject started any of the training, and also just before they received LPS. So there was no inflammation yet, but we've tested whether there already was an effect by just having followed the training for 1 week to 10 days. And here we saw that the pressure pain actually was decreased in the cold exposure group. So these subjects that were trained only in the cold exposure, they experienced less pressure pain after the training compared with before the training. For the electrical pain, we didn't see these effects. So this, again, I have to stress, this was not during the breathing exercise, but just before they started with the LPS and with the breathing exercise. There was no long-lasting effect of any of the training modalities on the electrical pain. But we saw strong effects on the cold-induced pain, and we saw those in the cold exposure group and in the combined cold exposure and breathing group. So also, if you have been training in the cold for a week or 10 days, it seems that you can better withstand the ice bucket challenge with your hand. You last for longer. You have less pain. Your VAS is decreased. So you might think, very nice, all these effects in healthy volunteers, but what about patients, of course, because that's ultimately what we, of course, want to show. And we did one small study together with the Amsterdam University in patients with Bechd's disease. So it's a certain form of arthritis, and it often manifests at a young age in otherwise relatively healthy people. And we thought that's a good group to test this intervention, because we can it's a relatively safe population with few comorbidities, and that's why we wanted to do the study in these subjects. And we also know in these subjects that these biologicals which antagonize cytokines, such as anti-TNF, for instance, they are very effective in this disease. So what we did is the patients followed the Wim Hof training, and this time in all 3 elements. So it was the full package, and Wim Hof and his certified trainers provided this this training. And first of all, of course, we looked at safety. I won't bore you with all the details and the adverse events, but we didn't see any difference between patients who followed the training and patients who were not following the training. It was actually a crossover study, so there were the same subjects, the same patients, but there was a period in which they practiced the training and a period in which they just did their lived their normal lives without the training. But we saw no differences in any no relevant side effects due to the training or related to the training. So if and these are more the disease measures that we did. So we measured a bunch of things and this is the trained group. And as you can see, for instance, ESR, which is a measure of inflammation in the blood, was significantly lower at the end of the training period compared to at the 8-week mark, after 8 weeks of training, compared with the baseline situation. And this was also true for ASDAS-CRP, which is a combination of a blood biomarker and a scoring list of swollen joints and symptoms, and also the BASDAI, which is also a similar endpoint. We also looked at physical, psychological functioning. Oh, this is, sorry, physical functioning, which is also improved after 8 weeks of training. And also the mental score of the SF-36 questionnaire, which you might know, I think, was significantly better after 8 weeks of training compared with the period before the training. And if we look at the control group, actually control period, we didn't see any significant difference at the 8-week mark compared with the baseline period. So I think we can conclude from the patient study that the training program, at least in these patients, it's safe and it's feasible. And we found indications that it mitigates the inflammation and the disease burden in general. But it's a small group of patients. Of course, it's a very preliminary study, and the design is suboptimal because it's a crossover design. And ideally, of course, you want to do a randomized controlled study where you just randomize patients to either the treatment or a sham treatment or no treatment at all. So the design is suboptimal and might be, has room for improvement in the future, I think. So yeah, that basically brings us to the end of my story. We did a lot of other stuff with Wim Hof. Within these studies, for instance, we measured metabolomics. There are some we had a paper on that. We also looked at like more psychological aspects, but for today I wanted to get more into the inflammatory and pain results that we gathered during these years that we studied this method. With that, of course, it's not something that you do alone, these kind of projects. And so we worked with a lot of other groups and other researchers, and they're all acknowledged on this slide. And with that, I'd like to thank you for your attention. And I'm, of course, very happy to take any questions.
Thank you so much. It's just such an amazing wealth of findings and amazing methodology. So I think people should unscreen, say hi, introduce yourselves in the chat. We have amazing researchers here, many PNI researchers. Some use endotoxin. And yeah, let's have questions. Maybe Eric, you could run the Q&A.
Yeah. Hey, yeah. And Matthijs, thanks for joining. Eric, right there. I'm really excited to have you here. I know. Let's see. Anybody have any questions here? I think Aoife, I'm going to put you on the spot.
Oh,
Hi, yeah, this is super interesting. I'm wondering if you could say more, or what do we know about the long-term outcomes of people who engage in these practices?
Yeah, that's a very, very good question, and of course, I get that many times, and the honest answer is we really don't know actually at the moment. So we, as I showed you, we studied a very acute model of inflammation. So I think the studies that show no real studies have looked at longevity, I think, yet of this method. I think what we can say about this is that at least that we also showed, for instance, that this adrenaline increase that you can mount this adrenaline increase not, for instance, not twice on one day. So it does. We also did a study in which we let them practice the breathing method for twice in a row, actually, with a couple of hours in between. And then we saw that the second time they were not able to mount this adrenaline response again. The thing is that the next day they were able to do that. So I think it's a bit of like maybe an exhaustion of your of the adrenaline, the adrenergic system there. But what I know at least is also from anecdotes actually from patients who use this method, because of course it's not a real established method to treat autoimmune disease, but there are many patients with Crohn's disease, with rheumatoid arthritis, who do this. And those people do the breathing exercise once per day, and they also try to do like the cold showers or cold immersions also every day actually. So I'm not sure whether like you can do this for a week and then stop, and then for my gut feeling I would say that you have to maintain it to keep dampening this inflammatory response. I think you cannot, if you stop it, I think the effect will wane pretty quickly, at least on our endpoints.
And did you see any evidence of like rebounding effects, or like if this is a stressor, could it be harmful over time?
Yeah, that's a good point as well. What we did a small study, is not published yet, where we measured hair cortisol. So cortisol, the stress hormone. You can also measure it in hair, and you can do like a time series then. So you can say, okay, I take a segment of hair, and that corresponds to like 1 month ago, 2 months ago, 3 months ago, everything. And we did that in a small group in our studies, in a subgroup actually, because some of the guys were bald, so we couldn't do it. And we saw actually that the long-term cortisol decreased due to training. So, and I talked a bit to endocrinologists and people about this, and they say it might be that if you like condition yourself to frequent stressors, you might actually, it might be a negative feedback, and you might dampen your long-term cortisol, because that's what you measure in the hair cortisol. So, but I'm not an endocrinologist. I'm not an expert totally on that. So that could be a mechanism at play. But at least it is an observation that we found that the long-term cortisol seems to decrease instead of increase. Although at first we also had this hypothesis that it might increase. So.
Yeah, thank you so much. I'm sure there's other questions waiting.
No worries.
Yeah, really cool work. Yeah, thanks, Aoife. Yeah, I have a quick question. Has this evidence been kind of like replicated in models of like if you inject norepinephrine or epinephrine and it produce like a equivalent spike? Do you see the same anti-inflammatory effect?
Yeah, that's actually the case. So especially we also did studies, because I'm an ICU researcher normally. Yeah, so in the ICU we use a lot of noradrenaline. It's like a vasopressor, of course, and it's a first-line vasopressor actually used in the clinic. And because I did these Wim Hof studies, I thought, okay, if adrenaline is such a potent anti-inflammatory agent, maybe noradrenaline is as well. And actually it is, because we done the studies and we saw that if you just infuse noradrenaline, even at quite low doses actually, you enhance IL-10 and you also dampen pro-inflammatory response. So this is definitely the case, yeah.
Cool. Yeah, thank you. That's really, really interesting. I know you talked a lot about like how quickly the spike falls. Is that crucial? Like, can you give, like, I imagine people with chronically elevated adrenaline or noradrenaline probably don't have low inflammatory markers. Is that the case? Is it like, does it need to be a quick spike and come down, or because it seems like chronic stress has the opposite effect?
Yeah, that's my gut feeling as well. And of course, we know that chronic stress is actually bad for autoimmune diseases. So I think you're right there. And I think this has been shown in animal models as well, that if you have long periods of adrenaline infusion, then it has it has not this anti-inflammatory effect anymore, and may even have a pro-inflammatory effect. So I think. And it's funny that Wim Hof kind of like felt this somehow, that you have to do this breathing exercise like half an hour or maybe 3/4 of an hour on a day, and that's it. He just said, okay, only do it once per day in the morning. That's it. Don't do it repeatedly on a day, and it's also quite exhausting. So I wouldn't recommend it as well. But yeah, he kind of knew in some way that this was the way to do it. But how he knew, I don't know as well.
Yeah, very, very cool. Thanks.
Sorry.
Welcome.
Sorry. Hi, Dr. Kox. So nice to see you here. Really excited that you were able to join us today. Alyssa mentioned in the chat, we've been looking at transcriptional pathways that we see changes with the Wim Hof method. And so I've been really interested in this and excited to see your talk. And I actually wanted to say one of the questions Alyssa put in the chat real quick, and then I have another one, just around if with the PNAS findings, the inflammatory response was more due to like a training effect or something, or more like the acute adrenaline effect there.
Yeah, I think the anti-inflammatory effect, you mean, right?
Yes.
Yeah. I think it's more the acute breathing effect that we saw in the second study. Of course, we saw the major effect due to this breathing exercise. And we also saw actually that they didn't need long training. So also for the increase in adrenaline, a very short training as instruction actually was sufficient to elicit this adrenaline increase. So it's also in the latest in the psychosomatic medicine paper that this very long training, at least for the adrenaline increase is not necessary. So if we just did a 2-hour instruction, and then you also already saw that these guys could do the breathing exercise quite well and also increase their adrenaline to these high levels. So I think that it's not necessary to do this long training to achieve this acute effect, at least.
Okay, that kind of leads into my next question, which I think is maybe less relevant now, but I'm curious your thoughts. You were mentioning some of the psychological factors that you'd looked at, and something that's been in the back of my mind around Wim Hof method is just the idea of these panic responses that our bodies have to cold and the that kind of breathing, right? And just what the experience of having that kind of response and being able to manage it and kind of control the interior experience of that is. And so I'm curious what kind of psychological findings y'all have found.
Yeah, I think the study the psychological study was done by of course by another group, because we are not the experts, and they looked a lot at neuroticism and all kinds of those kind of things, and expectations as well. I think they showed clearly that the expectations mattered a lot. But in my own experience, I think this method is really empowering for people, right? That they like exactly what you said. Normally, when you go into this cold ice bath, you panic and you like freeze, and you start breathing very superficially and all these kind of things. But what I noticed when these guys were trained, that they were able to really control that, and I think that gave them a sort of like a very mighty feeling of being able to control something that it's quite extreme actually. And I think for patients that could be something that's very important as well, to also to do something about your own disease burden. That is something that is I think is really valuable. But yeah, of course, that's all a bit of speculation there. And
Sure.
yeah, I'm not into all the details of the psychological study, so I have to.
Okay.
Yeah, it's a long time ago actually, but it's out
No
there.
worries.
So yeah,
Awesome. Thank you. Thanks for giving me your thoughts there. I was really curious. So I appreciate
no
it.
worries.
Alyssa had some stuff in the class. I guess I also wondered, I don't know if this was touched on already, so I apologize if it has, but you know, the training, like what we're really interested in, you know, is, you know, this is a tool, but also potentially as a lifestyle, right? And can we kind of change the set point for people physiologically? And maybe it's the autonomic nervous system specifically, maybe it's kind of something about the dynamics with the immune system. But do you see this as a potential way forward for kind of, you know, changing the overall health or someone's resilience in the long term biologically?
Yeah, that's also interesting because people often ask me, like also healthy people, should I do this method? And then I say, yeah, I don't know, because what we have shown that it's like it can be of value to if you dampen your immune response for certain groups of patients, actually, I think. And like whether this method has like generalized health benefits, I don't know actually. It's a very good question. And I think it has to be, hopefully it gets answered in the next decade or something. And I think you guys are also doing an amazing job on that to look at many other aspects of the effects of the training. But so we are, of course, I'm also working at the ICU department. So we're really focused on disease and like what we can do to help ill patients. So if it's like a generalized lifestyle that is beneficial for everyone or for a large group of people, it's very hard to answer that question. Actually, I think, yeah, as I say, at least in the US and in the Netherlands, it's a free country, so everybody's free to do what they want. But people often ask me, I think it's also in the chat, that if I do it, and I don't, but that's mainly because I like to keep a bit of distance between like what I research and like what I do in my private life. And as far as I know, I don't have any chronic illness or something. So that's of no, at least no autoimmunity. So yeah, I don't do it, but I might when tomorrow I wake up and I have swollen joints and I have rheumatoid arthritis. I think I'll try
it. Yeah, when we were doing our study, I did it one time, and I was like, "Whoa,
okay, yeah, I did
definitely
it once
definitely does
well.
something, that's for sure." But I don't know if I could make it a habit. Yeah,
it's quite impressive, right? If you do the breathing exercise.
Yeah, for sure. Anything I saw Aoife came on, so I don't know if you had another question or because then we have like 1 minute left.
Speaking of that, I mean, have you heard about any cases where people had a really bad acute outcome?
Yeah, of course. I know. I don't know whether you're aware of like the lawsuit that's running against Wim Hof at the moment in the US, actually.
No.
You're not? Okay. He's actually been sued by a lawyer from the US whose daughter died from shallow water blackout. So shallow water blackout is a well-known phenomenon that if you do this kind of hyperventilation exercise in water, it's very, very dangerous because as you may have experienced yourself or in the study, that if you do this breathing technique very strongly and profoundly, then you can lose you have some loss of conscious sometimes. And that's why we also in the video you saw that people were lying down on mats when they did the breathing exercise. And then nothing really can happen because if you kind of lose some consciousness, you're still lying down and you wake up and then it's fine. But you can imagine when you're in the water, and this is also a technique used, a similar technique is used by deep sea divers who do this hyperventilation and then can go under for minutes to dive for pearl divers and those kind of people in Asia most prominently. And there we but you can imagine if you pass out in the water and then you take a deep breath underwater, then your lungs are immediately filled with water, and it can be fatal. And that's what happened to this girl, actually, who 70-year-old girl who died, or at least that's what they suppose happened. And her father was a lawyer, and then he also gathered other cases of similar cases that are supposedly there. And in the Netherlands, there's also some cases that are related to this, or at least, they think that this had those people were at least practicing the Wim Hof method in their lives, and then they were found in the water. So this is a bit of I think he's sued for like $67 million actually, and he has to come before court. So that's a bad situation actually.
Yeah.
And we warned him a lot about these potential risks, and he also altered his videos to warn for this, so that's actually good. But there are old videos of him, like before we knew more about the method, and he himself is doing it in the water, so that's very unfortunate. So that's now being used as kind of evidence that people might have took inspiration from those videos.
Yeah.
So yeah.
Well, thank you.
I'd be
Amazing.
very curious to look at the RNA-seq results. Actually, do you have any? I think Ryan, you mentioned that you were doing these analysis, but is there anything?
Ryan, do you want to share? Why don't you want to share?
Yeah.
I know that I just want to say we're at time, so people have to go. Totally understand.
I have
So
Thank you,
for doing this,
everyone. Great turnout.
but you can definitely
for the
answer
summer.
this.